Welcome to lecture 14. We're back in chapter 9, Patterns of Inheritance, part 2. So during the last lecture, we talked about Mendel's laws. During this lecture, we're going to talk about variations of Mendel's laws. So during the last lecture, all of our offspring looked like one of their parents. So if we had a red flower and a white flower, all of their offspring were either red or white. But that's not always what happens. There's actually a couple of different possibilities for something in between that. So the first one is known as incomplete dominance, which is some kind of intermediate phenotype. So when Mendel did his P crosses, they always looked like one of the two parental varieties. That's called complete dominance. So if a red flower and a white flower make only red flowers or white flowers, that is complete dominance. Right? But sometimes you can cross a red flower and a white flower and the offspring actually come out pink. So in the F1 generation, all of them come out pink. This is something called incomplete dominance. So a red flower and a white flower makes a pink flower. That is incomplete dominance. Notice that this pink flower is heterozygous. So it still has the big R, it still has the little r, so the big R for red, the little r for white, it still has those alleles. But when it is heterozygous, when it has both the big R and the little r, the phenotype actually displays as something in between. That is incomplete dominance. But then, take a guess. What do you think would happen in the next generation? So this hybrid, this F1 hybrid, still has big R and little r. So if we were to cross two individuals from the F1 generation, what would their offspring look like? We'll take a look. It turns out that in incomplete dominance, the initial colors still exist. So in the F2 generation, since the F1 generation was heterozygous and is still passing on big R and little r, the offspring that are homozygous dominant, that are big R, are still going to turn out red. Then we have our heterozygotes, which are going to turn out pink, and our homozygous recessive individuals that will still turn out white. So in incomplete dominance, any heterozygotes are going to be some kind of phenotype in between the dominant and the recessive phenotype. So for some characters, the appearance of F1 hybrids falls between the phenotypes. Again, this is called incomplete dominance. And there's something different then called codominant. So in incomplete dominance, heterozygotes showed something in between the two parents. In codominance, they're going to show both the dominant and recessive trait at the same time. So if we're sticking with our example of the color of flowers, in incomplete dominance, the heterozygotes resulting from red and white were something in between. They were pink. In codominance, they're going to show both. So in codominance, if you mixed a red and a white flower, the offspring, the heterozygous offspring, would be both red and white at the same time. So imagine a flower that has streaks of red and streaks of white. If it's showing both at the same time, that is codominance. And the main example we're going to use for codominance is actually human blood groups. Because human blood groups, you can be A, you can be B, you can be AB, or you can be O. This is because of codominance. So let's take a look at codominance in human blood groups. So although each individual carries at most 2 different alleles for a particular gene, in cases of multiple alleles, more than 2 possible alleles exist. So while you only have 1 allele from mom and 1 allele from dad, it's possible in the example of human blood groups to get an A, a B, or an O. So there's actually 3 different alleles possible. You're only receiving 2 of them, but there are 3 alleles possible that you would get. So again, we're using the example of the ABO human blood group. So A, B, AB, or O are the phenotypes here. So the floor blood groups again are A, B, and O, resulting from a combination of either the A allele, the B allele, or the O allele. So A and B are both expressed and heterozygous individuals, making both alleles co-dominant. So A and B are dominant. O is recessive. Since A and B are both dominant, we call them co-dominant. So here's what that looks like. So on the left, our blood group, this is our phenotype. Your phenotype, your blood group can be either A, B, AB, or O. Now let's take a look at the genotypes. Now you'll notice that we use the letter I here. And so if we have a capital I, that means it is dominant. We do this because the blood groups are co-dominant. It's to remind you that A and B are co-dominant. So we have big I with either the superscript A or the superscript B representing, so big I A or big I B is either A or B. The big I just represents that it's co-dominant. And then O is expressed by a little I to show that it's recessive. So basically little I is the same thing as O. IA is A, IB is B. So if we take a look at our phenotype here for blood group A, right? So what we're showing is blood group A. Our genotype could be either IAIA or IAO because O is recessive. So if you have an A and an O, the A is going to show because it's dominant over O. So blood group A has a genotype of either AA or AO. For blood type B, the genotype is either BB or BO. Now for AB, this is where the codominance comes in. A and B are both dominant, they're co-dominant. So the AB blood group means you have both the A allele and the B allele. And since they are co-dominant, they are both going to show. So you have both A and B. O is recessive, so the only way to have O-type blood is for your genotype to be OO. Now if you take a look, you can actually physically see the difference here. So blood groups are defined by different types of carbohydrates. So there's a carbohydrate A, these little spiky ones. There's a carbohydrate B, which are these little circles. And here, if you take a look at AB, you'll see the codominance. If you are AB, you have both carbohydrate A and carbohydrate B, and they both show up on your red blood cells. So there's the codominance. Both of those alleles are showing because they are codominant. And then for O, you have neither of the carbohydrates. When we're in lab, we actually perform an experiment for this where we use antibodies that are present in the blood, and we'll put little drops into fake blood in order to see. And so if antibody B is present, we can put a drop in there, and any blood that has a B in it will clump. Any blood that does not have a B in it will not clump. For here, we put in antibody A, so any blood that has an A in it will clump. AB doesn't have any antibodies, and so none of them will clump. And then O actually has antibody A and antibody B, so anything with an A or a B will clump. So basically when we do this, if we put a drop in and only the B and the AB clump, we know they're blood group A. And you can see the pictures here. We can actually tell the blood group by dropping in the antibodies. And then here's that flower that I promised you. So here's an example of codominance where we have a red parent and a white parent, and it makes this nice red and white offspring. So again, that is codominance. So make sure you keep those two terms straight. If a red parent and a white parent make a pink parent, that is incomplete dominance. If a red parent and a white parent make an offspring that is both red and white, that is codominant. And a great example of codominance is human blood type. All right, so a single gene may actually affect many phenotypic characters. This is known as pleiotropy, when one gene influences many different characters. Sickle cell disease is an example of pleiotropy in humans. So sickle cell disease affects the type of hemoglobin that's produced and the shape of red blood cells and causes anemia and organ damage. Sickle cell and non-sickle alleles are codominant. Carriers of sickle cell disease have increased resistance to malaria, though. So if you take a look at the top, an individual is homozygous for the sickle cell allele. They produce sickle cell hemoglobin. The abnormal hemoglobin crystallizes, causing red blood cells to become sickle-shaped. That's why it's known as sickle cell anemia. And sickle cell anemia can cause damage to organs like kidney failure and heart failure. It can also cause other things like pain and fever and joint problems because the red blood cells are this sickle cell shape. And a single characteristic, so in the last example, we had one gene that controlled many characters. It's also possible for one character to be influenced by many genes. So an example of this is human skin color. So human skin color is a result of what is known as polygenic inheritance. So a single phenotypic character results from the additive effect of two or more genes. So human skin color is actually controlled by a lot of different genes, which is why there's such a large variation in human skin color, because it's controlled by so many different genes, and there are so many different options for those genes to show. So if you take a look here, you could do a really complicated cross here, but this is mainly just to show you that there are so many different genes that affect skin color that it has all of these really different beautiful options that we can show. So here's an example of pleiotropy. So a single gene affects many different characters, right? One gene going to many different characters. Or in polygenic inheritance, our second example, this is where we talked about human skin color, we can have multiple genes that all come together to affect one character. So the environment actually affects many characters. I think earlier I mentioned something about identical twins, like way back in one of the very first lectures, because we have identical twins where their DNA can be the same, but they don't always look exactly the same, and part of that is because of the environment. So many characters result from a combination of both heredity and the environment. For example, skin color can be affected by exposure to sunlight, and heart disease and cancer are influenced by genes and the environment. So take an example, oh, look at how cute they were back in the day. Identical twins, right? Cole and Dylan Sprouse here are identical twins, but they actually look a little bit different. And even though they have, are they identical twins? I don't know. But even identical twins who have the same DNA can be affected by the environment to look a little bit different. All right, so the chromosomal basis of inheritance. So think back to, was it last week we were talking about chromosomes? So you know about chromosomes. Let's talk about the chromosomal basis of inheritance. So chromosome behavior accounts for Mendel's laws. So there's the chromosome theory of inheritance that states that genes occupy specific loci or positions on chromosomes, and chromosomes undergo segregation and independent assortment during meiosis. If you're a little shady on this, look back at the last lectures, because I explained these things in the lectures from last week. So Mendel's laws correlate with chromosome separation in meiosis. There's that law of segregation that states that pairs of alleles separate from each other during gamete formation via meiosis, and the law of segregation depends on the separation of homologous chromosomes in anaphase 1. The law of independent assortment states that each pair of alleles sorts independently of other pairs of alleles during gamete formation and depends on alternate orientations of chromosomes in metaphase 1. So the different alleles are going to sort independently of other alleles, right? Just because this allele here doesn't affect the other ones. And then remember in metaphase, that's where our chromosomes are meeting in the middle, they're lining up in the middle. And alternative orientations exist, so they could line up this way, they could line up this way, right? Producing variability. So let's take a look here. We have all yellow round seeds up here in the F1 generation. Take a look here. We could have all the reds on the left and all the blues on the right, or we could have a little combo going on here. These are two equally probable arrangements of chromosomes at metaphase 1. Notice then that when we go through meiosis though, that depending on which way they line up is going to produce very different results for metaphase 2. Now the same thing can happen in metaphase 2. resulting in all these different combos of gametes at the end of meiosis. Here's what it looks like in your dihybrid cross, right? So these are basically the same thing here. This is just showing you how this happens. Hopefully you remember this, our 9:3 to 3:1 ratio from the last lecture. All right, so genes on the same chromosome tend to be inherited together. Now remember, there is some crossing over where during meiosis, sometimes chromosomes, homologous chromosomes will swap pieces. So this doesn't always happen, but for the most part, genes that are on the same chromosome are going to be inherited together because you're passing on a whole chromosome for the most part. So Bateson and Punnett studied plants that did not show the 9:3 to 3:1 ratio in the F2 generation. Now what they found was an example of linked genes, which are located close together on the same chromosome and tended to be inherited together. So if you take a look here at purple flower and long pollen, the prediction was that 9:3 to 3:1 ratio. But what they actually observed was something different. So they didn't see that 9:3 to 3:1 ratio between the color purple and having long pollen. They actually found that there were some combinations that were more likely than others that didn't follow that ratio. So why? The explanation is linked genes. So the closer genes are to each other on the same chromosome, the more likely they are to be passed on together. Because the closer they are, the less likely they are to be separated by crossing over. So if you have a chromosome and you have one gene at the top and one gene at the bottom, and it experiences crossing over with its homolog, it's much more likely that genes that are far apart are going to be switched out with the other homolog than if they're close together. Because the closer they are together, the more likely that they would experience crossing over together and they would be crossed over at the same time. So here's a close-up that just shows you they expected this ratio here. Notice on the right, 215, 71, 71, 24. What they actually got was 284, 21, 21, 55, which suggests that the purple and the long pollen are linked somehow. All right, so those two alleles are linked. So here's a close-up of the bottom of that. And it just shows here that we have these two genes, they're close together, so they're more likely to be passed on together. So again, crossing over produces new combinations of alleles. Crossing over between homologous chromosomes produces new combinations of alleles and gametes. Linked genes can be separated by crossing over, forming recombinant gametes. Now the percentage of recombinant offspring is the recombination frequency. And if you take a look here, they did an experiment with flies. So we had gray body, long wings, that's our wild type. We had black body and vestigial wings, so little tiny wings. And the offspring either looked just like the parents, so they were gray and long, or they were black and vestigial, or they were recombinant phenotypes, where they were gray from one parent with vestigial wings from the other, or black from one parent with long wings from the other. And you'll notice that the recombination frequency down here is 17%. So it doesn't follow that 9:3 to 3:1 ratio that we see. And the explanation here are the linked genes and crossing over. So again, the closer they are, the more likely they are to be inherited together. But sometimes, take a look here, sometimes crossing over can still separate them. And so that is what is happening here, is crossing over in some of these gametes has switched. And so, well, here, these genes could be linked, right? Gray body, long wings, they're located close together. So a lot of them will still have gray body, long wings, or black body, vestigial wings, but crossing over can happen. And so we can switch the type of wings, or we can switch body types sometimes. Those are the recombinant phenotypes, where we get one from mom, one from dad. And geneticists can use crossover data to map where genes are located. So when examining recombinant frequency, Alfred Sturtevant, one of Morgan's students, found that the greater the distance between two genes on a chromosome, the more points there are between them where crossing over can occur. So recombination frequencies can be used to map the relative positions of genes on chromosomes. So the closer genes are together on a chromosome, the less likely they are to be separated by crossing over. The farther apart they are on a chromosome, the more likely they are to be swapped during crossing over. We're able to do some math on this and use that, the amount of times we see crossing over, the probability that they're separated during crossing over, to show where these are located on a chromosome. And we call this type of diagram that we produce a linkage map. So we can see here, the blue and the orange are less likely to be separated than blue and black because they're closer together. So we can actually take a look at how often they are separated to guess the order. So say we know there's a black allele, we know there's an orange allele, we know there's a blue allele, but we don't know which order they're in on the chromosome. Well, now we can look at the math. We find that the blue and the black are separated about 17% of the time. But black and orange are only separated 9% of the time, and orange and blue are only separated about 9.5% of the time. So since blue and black are separated more often, we know that they must be farther apart than either blue and orange or black and orange. So we can do a little math here. We can figure out approximate locations of the genes here based on the recombination frequencies, the percentage of times that they are separated during crossing over. So here's an example in flies. So we are actually able to map where these genes are located based on how often these different traits occur. So red eyes and these Aristae here are pretty far apart, so we would expect that these would show a higher recombination frequency, a higher amount of times that we see crossing over. We see pink and blue are fairly close together, so we would expect that eye color and wing color would probably stay together. All right, so now let's look at sex chromosomes and sex-linked genes. So chromosomes determine sex in many species. In mammals, including humans, the Y chromosome has a crucial gene, SRY, for the development of testes, and the absence of the SRY gene directs ovaries to develop. So I mentioned in one of the previous lectures that if you have a Y, you are male. If you don't have a Y, you're female. So that's the difference. It doesn't really matter about the X's, it's the Y that's the difference. And this is Y, WHY, because the Y chromosome has that SRY gene that codes for the development of testes. So you either have the Y and it tells you to grow testes, or you don't have the Y, it doesn't tell you to grow testes, so therefore you grow ovaries. Grasshoppers, though, are a little bit different. Grasshoppers, roaches, and some insects have an XO system in which O stands for the absence of a sex chromosome. So females are XX and males are XO. So again, in humans and most mammals, if you were XO, you'd still be female because it's the presence or absence of that Y chromosome that determines your sex in mammals. It's different in some insects. So they both have X's, just like in humans and mammals. But if you have a second X, that means you're female. If you don't have a second X here, that means that you are male. So a little bit different. In certain fishes, butterflies, and birds, the sex chromosomes are Z and W. So males are ZZ, females are ZW. Some organisms actually lack sex chromosomes altogether, and most ants and bees, sex is determined by chromosome number. So females develop from fertilized eggs and are diploid. Males develop from unfertilized eggs, so they are fatherless and they are haploid. So if you take a look, these are 3 systems of sex determination that we just talked about that are different than in most mammals. So we have our XO, for insects, ZW for some of our birds, chromosome number for some other insects. In some animals, environmental temperature can determine sex. So for some species of reptiles, the temperature at which the eggs are incubated during a specific period of embryonic development determines whether the embryo will develop into a male or a female. An example of that are sea turtles. So if sea turtle eggs are incubated at a certain temperature, above a certain temperature, they're one sex, below a certain temperature, they're the other sex. Now, one of the problems of this system is that as the Earth continues to heat up due to climate change, once we reach a certain threshold, all sea turtles are going to end up being one sex. So we might end up with, you know, all one sex of sea turtles, and then they can't reproduce. So that's a little side effect that we've never thought of. Oh, cute little sea turtles. All right, so sex-linked genes exhibit a unique pattern of inheritance. So sex-linked genes are located on either of the sex chromosomes. The X chromosome carries many genes unrelated to sex. The inheritance of white eye color in fruit flies is an example of an X-linked recessive trait. So the white eye color in fruit flies is actually found on the X chromosome, one of the sex chromosomes. So there you go, red or white eyes. So this shows you a little cross here. We can have XR, XR here. So this one is homozygous dominant for red eyes, mixed with a male who has just the one X. So it's just the X little r, which means when we mix them, we get these different combinations down here, but all are going to be red eyes. If we mix that generation, we end up with 3 that are red, 1 that has white eyes. There's some different combinations here. Now notice, since that eye color is only on the X chromosome, and males only have 1 X chromosome while females have 2 X chromosomes, that our crosses look a little bit different than we saw in Mendel's laws, because the sperm from the male could be passing on a Y chromosome, which is not going to have any effect on eye color. So if the male is passing on a Y chromosome instead of an X, it's not going to affect eye color, and eye color is only going to be affected by the mother. However, if a male passes on the X chromosome to the offspring, that would have a gene for eye color. So when the male passes on an X, then it does affect the eye color of the offspring. So one of the important connections to this is that human sex-linked disorders affect mostly males. So think about why that is. Why is it males that mostly show sex-linked disorders? Well, think about, if you're thinking about chromosomes, what it means to be male and what it means to be female. Females have typically 2 X's, and males only have 1. A lot of genetic disorders are recessive, so you have to have 2 copies of that recessive allele in order for that disorder to show. So females who typically have 2 X's have 2 chances to get a dominant allele. So they might get the recessive allele from one parent, but they have another X. They have another X that could have the dominant allele. Males, though, only have 1 X. So therefore, if that 1 X has that recessive allele, they don't have another X to help them out. So sex-linked genes are located on either sex chromosomes. The X chromosome carries many genes unrelated to sex, right? We just looked at this. Most sex-linked human disorders, like I said, are recessive. And because of this, are seen mostly in males. Because a male only receives a single X-linked, they only receive a single X. So therefore, a male that receives a single X-linked recessive allele from his mother will have this disorder. But females have to get it twice. So males only have to get that recessive allele once. Females, since they have 2 X's, have to get it twice. So it's much less likely that a female will get both recessive alleles on both of their chromosomes. So recessive and sex-linked human disorders can include hemophilia, red-green color blindness, muscular dystrophy. And we've actually seen this. This is an example of one of the ruling families over here in Russia. And so these royal families are actually rather inbred. A lot of royal families are very inbred. There's a lot of like cousins marrying cousins to keep the bloodline intact. And so if you look back through history, a lot of royal families actually probably had these disorders. Since they are inbreeding so much, there's a much higher chance of passing on the disease because much more of them have the disorders. All right, so you should now be able to describe the inheritance patterns of incomplete dominance, multiple alleles, codominance, pleiotropy, and polygenic inheritance. Explain how sickle cell can be adaptive. Explain why human skin coloration is not sufficiently explained by polygenic inheritance. You can read a little more about this in your book. I briefly touched on it. If you want to get into the weeds, read about it in your book. Define the chromosome theory of inheritance and explain how the chromosomal basis of the laws of segregation and independent assortment result. All right, so there are some variations on Mendel's laws. That is the end of this lecture, and I will see you again soon.